right after that. Now we have the recording. Okay, awesome. All right, well, welcome everybody to the May installment of Forces at Play, our seminar series. This is a real treat today. We have Professor Arpita Upadhyaya. Arpita did her early studies and her undergrad degree at the Birla Institute of Technology in India. And from there, she moved to the US, where she worked on her PhD in physics at the University of Notre Dame. She worked with James Glazier, and at the time, she studied the biophysics of single hydra cells, which are really super cool organisms, both on the single cell and multi-cell assembly scale. Um, from those studies, then she moved on to conducting a postdoctoral fellowship at MIT, and she had one of these prestigious named fellowships at MIT, where she worked with Alex van Oudenarden. And at that time, she continued her work in the field of biophysics, and there she studied this really fundamental question, which is relates to actin polymerization, the forces generated by actin polymerization. And then she did some beautiful work there, and from there moved on to the University of Maryland, where she started her independent career with appointments both in the Department of Physics and at the Institute for Physical Science and Technology. And for her independent work, she's been recognized with a number of awards, including the Sloan Research Fellowship, and she has since been promoted, down the ranks, and she's really done some elegant work. And in her current lab, she really leverages her past experience and her expertise in studying the mechanics and the cytoskeletal forces in cells, and particularly in immune cells. And so this is going to be a really fantastic lecture, a beautiful fit to our theme here. And I would just end by mentioning that Arpita is really an avid photographer and hiker, and she loves to hike in exotic locations. So anytime you sit down and chat with Arpita, she'll have some really amazing stories of hiking in some remote regions of the world. And so I'm super excited to see your talk today, and I look forward to learning about forces in the immune system.
Thanks a lot, Khalid, for that wonderful introduction and for inviting me. I realized that my internet got cut out in between, but now it appears stable. So I'm going to try to share my screen quickly again. So let's see. So you see my screen? And do you see my slides properly?
Yep.
Okay,
yes.
Great. Great. So it's a great pleasure to be giving a talk in this excellent series, and thanks again for organizing this for the community and also inviting me. So when I started working on T cells, the beginning of setting up my lab in the physics department at UMD, I had not really envisioned that this new field of immunobiophysics would grow so rapidly, but it has really exploded over the years. And as Khalid mentioned, I like doing photography of macroscopic objects in my free time. So this is a view to what I feel is the portal into our building where our work is done. And this, of course, is a T cell that's also a portal into another world. So in my lab, I'm at the Department of Biophysics and also the Biophysics program at the University of Maryland, and the Institute of Physical Science and Technology. So in my lab, we're interested in how the cellular cytoskeleton and its regulators self-organize, define macroscopic mechanical properties of cells, and enable fourth generation for specific functional outputs. And a lot of these have been studied, very well studied in adherent cells when we were starting out, but immune cells are very rich system to ask some of these questions. And the cytoskeleton especially is very dynamic and over the scale from seconds to minutes undergoes extensive reorganization And it has its characteristic features which are very different from adherent cells. So even though a lot of the molecular players are similar, these provide a very interesting system to study the cell biology. And so over a number of years, we've been studying dynamics and mechanics of the cytoskeleton in immune cells and T and B cells as they relate to their functional outcomes and how these cells sense and respond to physical cues. So I like to think about our work in terms of molecular machines, thinking about how these machines work at multiple length scales and time scales. So the single molecule level, of course, you have enzymatic activity of receptor ligand interactions, for example, resulting in conformational changes, which can exert forces. And then at a larger length scale, you see sort of the mesoscopic assembly of cytoskeletal networks, which are these amazing self-assembly, self-organizing machines that themselves have their own mechanical properties, the viscoelasticity, and then they lead to force generation in cells. And here you can see sort of this rich dynamics of actin and microtubules at the T cell synapse. As these cytoskeletal proteins sort of polymerize and depolymerize, they push against the cell to generate forces. And we're also interested in somewhat larger length scales and studying cell-cell junctions. And this just shows an example of a CAR T cell interacting with a target cell and undergoing the downstream processes. And over a number of years, we keep thinking about and occasionally have been thinking about collective cell migration. But I like to show this movie because it shows the dynamics, kind of the cell-cell dynamics, and how the cell-cell interactions as well as the cell matrix interactions are important to to modulate the overall organization, even at larger length scales. So, for example, during tissue organization and development. So this particularly is a very interesting time to study the dynamics of these molecular machines and their functional consequences. There are many advances in molecular biology that allow us to label different components, perturb them in precise ways, and advances in super resolution imaging and also computational image analysis allows us to visualize and interpret these dynamics at unprecedented resolution. So in my lab, we kind of have been working on two different aspects. One which I'll talk more about, how does cytoskeletal dynamic shape changes and receptor signaling in immune cells. And more recently, we're also interested in what happens in the nucleus and how cells sense and transmit mechanical information into functional outputs. And I won't talk about this today, but maybe at another time, we're looking at high resolution imaging of dynamics within the nucleus, such as transcription factors and chromatin, and eventually interested in understanding how cells sense and transmit all of these diverse cues into different types of functional outputs and gene expression. So just a brief overview about T cells. T cells and also B cells and many other cells have specialized receptors on their surfaces. So let's think about the T cell receptor or the TCR. And receptor binding leads to activation at the molecular level and triggers the activation of the signaling of the TCR, which then leads to recruitment of other signaling molecules and formation of signaling microclusters. See, these microclusters are kind of the fundamental unit of signal amplification. And eventually, these lead to the activation of a large cascade of signal transduction pathways that then lead downstream to cytoskeletal reorganization and over longer scales, gene expression and T cell regulation. All of these components must act in concert in highly coordinated ways to lead to the cellular response. And now I'm going to show you this movie that maybe everybody has seen, but it's such a beautiful movie that it's hard to, oops, is it not playing? Interesting. Uh, well, I'm sorry this is not playing, but if it was playing, Can you hear me? Yeah.
Yeah, I can hear you. It's
Okay.
not playing, but
Okay. Um, but what you would see is this dynamic organization of the cytoskeleton as a CTL, a cytotoxic T lymphocytes interacts with the lymphoma target. So immune cells have this extremely dynamic cytoskeleton, which allows them to navigate a complex environment in the body. And over a number of years, a lot of prior work has shown that receptor-mediated signaling and cytoskeletal dynamics are tightly coupled and also interact in this bidirectional manner. So why are cytoskeletal dynamics important? So this extreme dynamic reorganization is an important feature of immune cell response. It's also critical for their function. So at the very basic level, these dynamics allow lymphocytes to spread and scan the antigen-presenting cell for antigens. These interactions between different cytoskeletal components act in microtubules, molecular motors, help the lymphocytes to adapt to their complex environment. And at the molecular level, we can see that the forces that arise from these dynamics may drive conformational changes of receptors, exposing signaling domains, leading to enhanced signaling and further signal transduction. Also, forces may facilitate assembly of signaling molecules, sort of this very tightly coordinated spatiotemporal organization into receptor clusters and mesoscale structures. And these are also important to drive the movement of these clusters then into larger scale organizations, which then mediate downstream signaling and longer scale gene expression, for example. So all of these dynamics and forces are important kind of from the molecular scale to the cellular scale. So since this is a series on forces, I wanted to give a little overview on what cellular forces might do in T cell activation. It has been, so as in, I forgot to point out a lot of these papers that and these are only a subset of the papers, but in the last more than a decade, there's been a huge number of studies in this very rich field. So it turns out that receptor binding alone may not be sufficient for triggering TCRs, and it led to the idea that forces are required to activate T cells. When we started working on this project many years ago, not a lot was known about this, and there were some theoretical ideas that proposed that forces may be important for T cells. But subsequently, there's been a lot of work that has shown, for example, that forces may directly lead to TCR activation with a certain threshold for receptor activation. And it's also been shown in this sort of seminal work that I cite over here from Cheng Zhu's lab, but also other labs and a lot of work in the recent past. The T cell receptor forms catch bonds with its agonist ligands, and then the signaling induction depends on the magnitude, duration, and properties of these forces exerted. And more recently, it's been shown that forces are required for T cells to kill cancer cells, allowing these cytotoxic T cells to target and eliminate cancer cells in the body. So with this background, just pointing out some questions that we've been focusing on in our lab in this area. So we've been interested in how the cytoskeleton reorganizes at the immune synapse during the activation of signaling, and how do these interact and generate forces, how they drive receptor movement and signaling, and more recently, how do these forces enable cells to respond to physical cues and modulate cell function? So I'll give you a brief overview of some of our previous work in this field, and then I'll talk about some of the newer work from our lab. So this is showing some very early slides when we just started working in this field. As you can see, the T cell and the APC form a contact, and if you wanted to study this in vivo, it's hard to get a very high resolution precise view of this interface, sort of the immune synapse, the T cell APC interface. So a number of labs pioneered use of planar substrates. I'll just show you the most basic one in the beginning. So we started out studying Jurkat T cells activated on anti-CD3 coded cover slips. And we first used sort of turf imaging to look at actin dynamics during cell spreading. I'll talk more about the different actin structures, but you can also already see that there's sort of this complex spatiotemporal organization of actin in the first few minutes of contact. And this is accompanied by the formation of signaling microclusters. Here you see ZAP70, one of the earliest signaling molecules labeled with YFP. And let me just see if, and in one of our earlier works, we also showed that the actin, the ZAP70 clusters, wow, so it looks like not all the movies are playing. Oh, it's playing. That actin polymerization appears to be initiated in these very early stages of contact and from the initial microclusters. So all of this shows that there's this rich dynamics and there's interaction between signaling and actin polymerization. So as you can see, the actin, if we look closer into the synapse, again, this is with the Jurkat T cell activating on an anti-CD3 coated glass surface, we see that the actin and myosin adopts a characteristic ring-like structure. And you can clearly see the two distinct structures, the peripheral sort of actin ring that shows the classic retrograde flow, and more internal ring, which is composed of these myosin arcs. You can see sort of these bundle-like structures. And this is seen more clearly with the super resolution imaging from the Hammer lab that shows these myosin arcs. And these are actomyosin structures, which are essential structures and features of the immune synapse. So a quick overview of, we wanted to understand the biophysical mechanisms that led to this global patterning of the actomyosin network, especially the formation of this ring. So we collaborated with Garrick Papoyan's lab, where they use median, the simulation that does coupled stochastic chemical of chemistry, as well as polymer mechanics. And so it's basically a chemical reaction diffusion dynamics that includes mechanics of actin polymers, as well as myosin and cross-linking proteins. So in that work, they considered two basic ingredients, actin retrograde flow by treadmilling and myosin contractility. So if there's no myosin and treadmilling is fast, the network just becomes a disordered patch. If you add myosin and slow down the treadmilling, the network just collapses. But then if there's a well-balanced composition of treadmilling and contractility, then you see that the network self-organizes into a ring-like structure. So the physical intuition is that for fast treadmilling and a given level of contractility, the ring state is an energetically favorable state that balances the bending of filaments with myosin contractility and stretching. So this gives us an intuition of some of the minimal ingredients for spatiotemporal organization of these elements. And this sort of opens up this possibility of conducting more complex simulations with, of course, many more of the ingredients that are found in a real system. As you can imagine, these dynamics of the cytoskeleton lead to forced generation of the synapse. So I'm just showing you very quickly some work early on in our lab when we first started working on this. And I guess we were among the first labs to be able to measure the forces generated by T cells. It took some time because these forces are actually very low compared to adherent cells. As you can see here, if you allow the cells to get activated on polyacrylamide gels that are coated with an activating antibody, the cells spread out. Here you can see the actin ring, and you can see the movement of the beads. So this is sort of the classic traction force microscopy and generation of traction forces, as you can see kind of on the footprint of the cell. These gels also are soft, so they can mimic the stiffness of antigen-presenting cells, and more on that in a few minutes. The stresses are similar levels to those that were shown to be able to activate the T cell receptor. And so it suggests that these cell-generated forces may be sufficient for T cell activation, and also that these forces are specific for TCR activation. And this early work I'm showing is in Jurkat T cells, but also around that time we found, and others, that CD4 T cells from humans also exhibit similar forces. Since then, there's been a lot of work on fourth generation in T cells, B cells, other immune cells, using a variety of different techniques, many of which you've already heard about in the seminar series and others, and you will hear more about them. So traction forces can be measured using, for example, PDMS pillars or polyacrylamide gels, as I showed you. As Khalid's lab and others have pioneered, you have DNA-based tension sensors. So while traction forces using polyacrylamide gels, for example, can show directionality of the forces, the spatial resolution is somewhat limited, on the order of a micron or less. With the DNA tension sensors, you can measure forces with much higher spatial resolution. And this provides a range of forces generated under given conditions. There's also been used by the biomembrane force probe by many labs, where this allows measurement of cell-cell interaction and also molecular level forces. From a lot of these, it's clear that the forces generated are sort of in the nanonewton range across the cell. They develop rapidly within minutes of cell activation. Actin cytoskeleton has been shown to be important for force generation, and myosin activity is important but plays a variable role. And so we did some work a few years ago trying to understand the role of microtubules in force generation, which was not as well studied. So what does the microtubule cytoskeleton look like at the immune synapse? So what I'm showing you is after a few minutes of synapse formation. I don't see the labels here, but what you see in green are EB3, so these microtubule tips. So once the centrosome polarizes the microtubule organizing center, it comes to the synapse, and you see a rich assembly of microtubule dynamics at the synapse. And it interacts with the peripheral actin ring, and you can kind of see that they're excluded from this very peripheral lamellipodial actin ring. We also see that the tip influx into a region, into this peripheral region, is associated with a slowdown. So what happens if you perturb the actomyosin cytoskeleton, and vice versa? What happens if you perturb the microtubule cytoskeleton, and how does it affect the actomyosin cytoskeleton? So what we found is, in this movie, as you can see, this is the normal cell. You have formation of the actomyosin ring. So when you introduce nicotinamide at low concentrations, so with low doses of nicotinamide, you have the microtubule network intact, but the tip dynamics are slowed down. So with these, under these conditions, you see the striking enrichment of this myosin ring after nicotinamide. And just as a comparison, when you inhibit myosin, of course, you lose this actomyosin ring. So this enhancement of the myosin ring, we found, was also associated with an increase of traction forces generated after addition of nicotinamide. And in this work, which I won't talk in detail, we found that this happens through the suppression of ROA activation, myosin bipolar filament assembly, and involves actin retrograde flow. So, and then we wanted to ask whether actomyosin forces have a reciprocal effect on microtubule growth and dynamics in turn. If so, it would point to an intricate feedback loop that maintains force balance at the immune synapse. So this work by Ivan basically showed that microtubule growth is slower in F-actin-rich regions, and also that So I wanted to point out in this movie over here, which is an ISIM movie showing that sort of different regions of actin polymerization. You see the retrograde flow at the periphery and these actin bundles more in the interior. So these bundles are formin mediated by the periphery is mediated by ARP2/3 mediated branched meshworks. ARP2/3 inhibition led to increase in microtubule growth rate. And we found that formin inhibition using small molecule inhibitors, we found that it leads to decrease in microtubule growth rates. So we also looked at the overall microtubule fluctuations at the synapse. This was actually the movie I showed at the very beginning, but you can see at the periphery, you have these really strong buckling of the microtubule filaments, but they're much straighter. And what we found that if you inhibit actin or myosin, you really get a strong reduction in these buckling and sort of fluctuating microtubule shapes at the synapse. So possible explanation for all of this is that formin-mediated, sort of in this case, formin-mediated contraction might keep microtubules more straight, while forces exerted by ARP2/3 networks might tend to bend the microtubules. And that's basically what we observed by looking at the overall microtubule dynamics at the periphery. So this gives you an overview that there's a crosstalk between the actomyosin and the microtubule cytoskeleton in T cells, and more work is needed in order to understand the mechanisms that underlie these crosstalks and how these different components, including the feedback between them, mediates an optimal force balance as well as signaling and function in T cells. I'll give you one brief sort of slide showing some work that we did using B cells as a model system. And the main takeaway here I wanted to point out is that cytoskeletal dynamics are important not only for force generation and changes in large-scale cellular morphology, but also at the more molecular scale, and they're important for spatial patterning of proteins, as well as the nanometer level dynamics of receptor molecules and signaling molecules. So this just shows some signaling, this single molecule imaging of B cell receptors, and just shows this wide range of diffusion coefficients. Without going into details, I'll just point out that the diffusion, the mobility of these receptor molecules are correlated with their signaling state. And using a mouse model with a conditional NWASP knockout, we found that this resulted in a slowdown of BCRs, abolished their centralizations. And other work, for example, from John Hammer's lab has shown that formin inhibition leads to a more scattered organization of TCRs. And as a result of this impaired clustering, the accumulation of signaling proteins into micro clusters that amplify signaling is further reduced. So these are just a few examples, but to point out that actin dynamics and its regulation is important for the spatiotemporal patterning and organization of receptors, co-receptors, and signaling molecules. With that, I will talk about some of our more recent work understanding how cytoskeletal dynamics and forces enable cells to respond to different physical cues. As you can imagine, immune cells can sort of see or sense a variety of different physical cues and chemical stimuli, antigen-presenting cells, and the sort of the microenvironment that immune cells are in have a range of stiffnesses. Often when these cells are soft, from a few hundred to a few pascals to a few kilopascals. The mobility that these receptors encounter because of antigens and ligands that are present on the APC membrane, which is more like a fluid membrane. And a number of work, including our own, has shown that ligand mobility modulates signaling in different cell types. Also, the topography is complex because the surface of antigen-presenting cells is not smooth, can have protrusions and ruffles from the nanometer to micrometer scales. And also there's a variety of cytokines that cells are subject to. So efficient signaling must involve adaptive responses by the cellular force-generating machinery. And we wanted to understand how does the cytoskeleton enable cells to respond to its environment. Very quick summary that a number of years ago, we looked at topography, collaborating with John Furcus at University of Maryland, using nano-fabricated ridges with variable spacing coated with anti-CD3. We can see that both the cell shape and the actin dynamics, as well as actin flows, are modulated by the spacing of these ridges. We also found microtubule dynamics are modulated by the ridges because the nanotopography aligns both actin and microtubule dynamics depending on the particular topography that, and so this has implications in terms of how cells might respond to different types of topography that they encounter in nature. We found again that ZAP70 signaling, basically the spatial organization of ZAP70 signaling was altered in T cells as well as in B cells. And these raised questions about what type of mechanisms might underlie this topography sensing and implicated curvature sensing signaling proteins that might lead to topography sensing by also leading to downstream activation of signaling molecules and cytoskeletal dynamics. But this is an area that requires future work. So in addition to topography, a lot of work has shown in the last many years that stiffness is a modulator of T cell function. And this I'm showing some of our earlier work showing, for example, that T cells are, the dynamics of T cells are different on different stiffnesses. This was with the Jurkat cell. Traction forces are modulated based on stiffness, and cells can adaptively increase their forces generated as the stiffness increases. Intriguingly, we found that on softer substrates, early signaling was sustained over longer time periods, while on stiffer surfaces, the signaling peaks at early times and then drops off. And again, these are some references in recent literature showing how substrate stiffness has been shown to modulate various aspects of T cell activation and also gene expression. So finally, I'll talk about some work recently that we did to understand how cytotoxic T lymphocytes respond to various chemical and physical cues with the goal to understanding how do they respond to stiffness of a cancer cell. So this work was led by very talented graduate students, Ashley Patney, who's recently graduated, and also very talented co-workers, Vishapdeep, Elizabeth, and Frank, and in collaboration with Dr. Zhengguo Zhao at UMD. Brief review about cytotoxic T lymphocytes. They're activated during interaction with antigen-presenting cells. Once activated, these naive CD8 positive T cells undergo clonal expansion and differentiation into effector cells. So effector cells can infect, can kill cancerous cells, as you can see in this amazing movie, through polarized secretion of lytic granules, which contain perforin and granzymes. Perforin forms pores in the target cell membrane, allowing granzymes to enter and trigger apoptosis. CTLs integrate different chemical and mechanical signals in their complex microenvironment to carry out their function. Particularly, cytoskeletal remodeling is important. And so cytoskeletal remodeling, as you see here, and the polarization of the centrosome towards the synapse is critical for this function. And the movie that didn't play earlier, that was, again, a lot of these amazing movies are from Gillian Griffith's lab, and that you can see over here. So centrosome polarization drives lytic granule transport, And also, there's this early stage depletion of actin, as you saw before with the Jurkat T cells, also in cytotoxic T lymphocytes, you have formation of this actin ring, and then which allows initial release of granules at the immune synapse. So this just again reiterates that T cells have to navigate these different mechanical and chemical cues, which lead from cancer cell softening, the ECM density, compressive stresses, fluid pressure, etc. And they must adapt to these physical cues to function effectively. So T cells also see a variety of different biochemical signals, and just give you an overview of the type of signals that we're using when we activate T cells. So classically, naive T cells are activated by sort of signal one, the antigen or the peptide MHC complex, and co-stimulatory signals such as ligand binding to CD28 receptors. So we'll refer to this as the two signal. More recently, inflammatory cytokines have been characterized as an important third signal, which we can refer to as signal 3 or 3SI. For cytotoxic T lymphocytes, these cytokines modulate and enhance the killing response. Our collaborator Zheng Guizhao has shown that CTLs activated in vitro in the presence of IL-12 have improved effector function. So as you can see in this in vitro killing assay, the 3SI CTLs are much more efficient in killing cancer cells. And also in this in vivo assay, where 2SI and 3SI cells were transferred into tumor-bearing mice, you see a strong reduction in tumor size in mice that received the 3SI cells, as you can see over here. So we spent a little bit of time trying to understand what the effect of this activation in the presence of IL-12, what does it modulate in terms of the cytoskeletal dynamics that we see in the immune synapse. We found very quick summary of this work, which was published. We found that 3-signal CTLs typically form larger synaptic interfaces with higher actin depletions and also, interestingly, reduced actin flows. They exhibit increased microtubule tip dynamics. And as you can see here, we're just looking at LAMP1 labeled granules, not the secretion itself, but we found that 3SI CTLs have larger number of granules, which are more dispersed over the synapse. These are some of the important differences that showed up when we looked at the synapse of 3SI versus 2SI CTLs. And so this led us to ask the question of what about the forces exerted by 2-signal or 3-signal activated TCLs? So our findings suggested that the forces exerted by TCLs might be different. And here I'm just showing you an example of the traction map as the cells are spreading out on the activating gel surface. And we find that the 3-signal CTLs exert greater traction forces. And this is consistent and with their exertion, with their observed enhanced cytolytic function that we saw. So just to summarize very quickly, we found that 3-signal activated CTLs showed enhanced actin depletion, more dispersed and more numerous lytic granules, low actin flows, which might lead to a more stable synapse and greater force generation. So now coming to the question of how target cell stiffness modulates cytoskeletal dynamics, forces, and function. So it's known that cell stiffness is altered under pathological conditions, and metastatic cancer cells, for example, have much smaller range of stiffness compared to normal cells. And this is shown in this AFM study, where cancer cells are shown to be much softer than their normal counterparts. Intriguingly, tumors tend to be stiffer than individual cancer cells. So how does target cell stiffness modulate cytoskeletal dynamics, forces, and function? So in order to study this question with some of our techniques, we used the polyacrylamide gels to activate 3SI. So we used 3-signal T cells on gels of different stiffnesses. So here I'll show you a movie where you can see, sorry, this is not, I wanted to play it over in. So this is the first few minutes, and let me just pause. You see that the actin of this is now a cytotoxic T lymphocyte, rapidly reorganizes into a ring, and here you see the granule accumulation. And over time, but very quickly, the ring closes, and you actually see the formation of these actin foci, which become more pronounced over time. Concurrently, the centrosome, which can be in any orientation with respect to the synapse, and here you see a centrosome that starts out at the top of the cell. Over a number of minutes, the centrosome repositions towards the immune synapse following contact with this activating gel surface. You can also see that the lytic granules coalesce, appear to cluster around the centrosome, and then are transported towards the synapse. To quantify this actin ring, we defined a depletion ratio, which is the ratio of actin fluorescence intensity in the center versus the periphery of the ring. What we found using these live cell movies, as you may appreciate, that the actin ring forms more rapidly on stiffer surfaces and compared to the softer surfaces. So this just shows this actin depletion ratio, which goes down, meaning the ring becomes stronger, faster on the stiffer surfaces. And we see the same thing here with the fixed cell images showing that at 4 minutes, you already have an actin ring on the stiffer surfaces compared to the softer surfaces. So the kinetics and strength of this ring are enhanced on stiff surfaces. The synapse morphology is also sensitive to stiffness. As you can see, this is fixed cell imaging taken at 15 minutes. You see the synapse area increases with stiffness. The cell deformation ratio, which is basically how much the cell deforms compared to how much it spreads, this is also increased. So the cell is more flattened on these stiffer hydrogels. We also saw that myosin light chain phosphorylation increases with stiffness, and and also early signaling, which I'm not showing over here. So next, we wanted to study how traction forces are modulated as a function of stiffness, given changes and modulation of both the actin and myosin cytoskeleton. So what you see over here is a movie showing the beads that are embedded in the gels, in this case, the lytic granules. This movie, you don't see the actin cytoskeleton, and you can see strong deformations of the beads. What we found interestingly, so this is the maximum intensity projection. What we found here, if you just see one slice at the gel surface, you see the strong sort of depression in the gel surface. So this indicated that there's clear and extensive three-dimensional deformations of the surface, and that the cells are exerting both tangential and normal forces. And we wanted to resolve both in-plane and normal forces. Wanted to point out that some recent work by Morgan Hughes, Dan Borslin, and co-workers have shown CTLs interacting with these soft particles, and they've also reported formation of these compressive synapses. So we wanted to look at this deformation at a bit higher resolution. So we used confocal microscopy to get sort of the 3D position of the beads on top of the gel surface, as well as a few slices below. And then we used a combination of particle tracking and correlation-based techniques to get a high-resolution displacement map. The displacement fields were denoised to remove large spurious movements. And then the 3D Green's function for the elastic half space was used to solve for the traction, both in plane and out of plane. So what we see from here is a movie showing the in-plane forces on the left, the normal forces as evidenced by the sort of the transparency. The blue, the darker blue shows more normal forces, and also the granules, as well as you can see with the displacement maps. Very intriguingly, the granules always tend to accumulate in this crater that is formed by the CTLs and appear to be localized to the edge of the crater. So we're doing further analysis to really quantify the correlation between the granules and the forces, but this just shows a snapshot of the in-plane normal forces and the normal displacements. We found that force generation, as expected, is dependent on actin networks, but it was interesting to see the strong effect both of formin inhibition with SMIFH2, which inhibits formins. As I mentioned, they are responsible for these bundled actin-myosin arcs. It led to a strong decrease both on the total parallel forces and perpendicular forces, as well as inhibition of ARP2/3, which reduced, again, both the tangential and the normal forces. And again, ARP2/3 is responsible for the branched meshwork at the periphery, and we use CK606 as an inhibitor of ARP2/3. We also found that CTLs exert higher forces on stiffer substrates, and this is just summarizing the data, showing at 1.5 and 6 kilopascal. It's been a little difficult to resolve the forces on the 12 kilopascal gels, but we're working on it. But generally, we see the same trend. You have increased forces on the stiffer gels, both parallel, and this is showing the root mean square traction and the total force exerted by the cell, as well as the perpendicular forces. So from then we wanted to examine how stiffness affects centrosome polarization as a function of stiffness. So we can do this both with live cells and fixed cells. So here I'm showing you fixed cells. You see the centrosome over here, which we identify by staining beta tubulin. And these are 3 different stiffnesses. And what we find is that activated CTLs display more efficient centrosome polarization on surfaces of higher stiffness, both in terms of the centrosome synapse distance, as well as the fraction of cells that are unpolarized. And you can see this also in live cells, just a couple of movies. Sorry, I don't know why some of the movies are not playing, but you saw a similar movie earlier, so you get the gist of that. We find the fraction of cells with polarized centrosome is higher on stiff gels. We also wanted to confirm with our assays and see how this affects, given that we see increased centrosome as well as granule polarization, how this drives differences in granule secretion. So we use a flow cytometry-based assay to detect granule release when CTLs were activated on surfaces of different stiffness, using LAMP1 as a marker of granule release upon activation. And we see that with flow cytometry, the increased LAMP1 fluorescence indicates increased granule release on stiffer substrates, as you can see over here, more strikingly so at 180 minutes. And then we've been working on some more functional assays, and I'll go over this very quickly to get to this question of target cell stiffness. So we can do this imaging-based assays at the population scale. Here you see melanoma cells, B16 melanoma cells, which is a form of skin cancer in the presence of CTLs. And as the melanoma cells die, they incorporate propydium iodide, which you can see in these red sort of spots over here. And these movies are over 18 to 20 hours. So in some preliminary data, we see that B16 cells plated on gels of different stiffness. So these are cells plated on 1 kilopascal, and here B16 cells plated on 12 and 50 kilopascal show different levels of killing, indicating cells that are grown on higher stiffness, which is indicative of increased cell stiffness, as I'm kind of quickly showing over here. Many different cell types have been shown to increase their own stiffness when they're grown on gels of different stiffness. So the So B16 cells, which are on higher stiffness, likely have increased stiffness, which is leading to an increased killing function of CTLs, as you can show over here. We're actually working on a number of different parallel assays, and maybe more on that in the future. But this was really interesting to see, but this is also consistent with some previous work in the literature. So in the last maybe few minutes, I don't know exactly how much time I have left. I'll just wrap up with some potential mechanisms. Yeah. Um, so to understand how T cells, so what might be the mechanism of this mechanosensitive behavior? And one of the key players, so of course, we have the TCR, which has been shown to be mechanosensitive, but then also more and more integrins, such as LFA1, have been shown to be mechanosensitive. And the application of force through these integrin ligand bonds might open up the integrin conformation and allow forces to be transmitted to the actin cytoskeleton. So we wanted to find out how the cytoskeleton helps, both integrins and cytoskeleton help CTLs respond to targets of different stiffness in order to regulate killing. And in recent work from many labs, and I just cite a few here, it's been shown that when LFA1 is blocked, it leads to reduced killing, and it's important for synapse formation, adhesion, for generation, as well as cytolytic function. Does this engagement contribute to the observed CD8 positive T cell mechanosensing? So again, the next few slides are very early preliminary results that we are following up on, but wanted to point this out. We also saw, as expected, that centrosome polarization itself does require integrin engagement. So you see a reduced fraction of centrosomes polarized when you block integrin on both but this happens on both stiffnesses. So these two plots show the soft, and then these two bar graphs show the stiffer surfaces. So it appears that integrin engagement, while important for centrosome polarization, is not obligatory for the stiffness-dependent response. We also found that cytoplasmic dyneins that have been shown to decrease centrosome polarization. We observed a modest decrease in centrosome polarization, fraction of cells, as well as the distance when we inhibited dynein with celiobrevin, but both in soft and stiff substrate. Interestingly, in our assays, blebistatin had limited effect on centrosome polarization on both stiffnesses. Sorry. And intriguingly, when we inhibited dynamic microtubule activity with nicotinamide, we found that it also inhibited centrosome polarization, but to a greater extent on the soft surfaces. We found similar results with inhibition of ARP2/3, which reduced centrosome polarization significantly on the softer surfaces and much less so on the stiffer surfaces, whereas formin inhibition reduced centrosome polarization on both stiffness. So with that, just wanted to point out that we are trying to understand like an overall model and the mechanisms for stiffness-dependent responses that we have observed. But it appears that on stiffer surfaces, there's greater force exerted both through TCR and LFA1 engagement, which results in enhanced signaling, increased actomyosin dynamics and contractility, increase in force generation, centrosome polarization, and also granule secretion and cytotoxicity. So some questions we'd like to answer are, what are the consequences of mechanosensing on gene expression, longer-term responses, and how other mechanical properties might affect CTL activity? With that, just a snapshot of some ongoing work. We've been also looking at the nucleus of T cells. This is a jerkat T cell, trying to understand how cytoskeletal dynamics modulate nuclear shape, mechanics, and vice versa. And then the role of the intermediate filament at the immune synapse. So this is a movie of Bimentin. And finally, just a snapshot of work we are doing in collaboration with Tim Lutkins at the School of Medicine understanding how CAR T cells function and figuring out this intriguing phenomena of trogocytosis and the role of CAR affinity. So with that, I just wanted to point out, like, overall, we have this picture of the immune cell cytoskeleton as a mechanochemical module, which appears to integrate both chemical and mechanical stimuli to drive CTL function at the level of synapse formation to target cell killing. And then this dynamics is apparent at the whole cell level, as we see from sort of this 3D movie of a cytotoxic T lymphocyte. And so our work and a lot of other work in the field has shown that these mechanical cues are emerging as fourth signal in immune cell activation. And there's a lot of work still to do. But with that, I wanted to thank all the people who made this work possible. Of course, I did not do the work that I just presented, though in my defense, I do help with data analysis. I'm thankful to have had an amazing group of people in my lab over the years who've been extremely energetic, creative, and hardworking. A lot of the work that I presented was led by Ashley, and then in collaboration with Vishwadeep, Frank, Elizabeth, but also point out all my current lab members that are working on projects that I did not have a chance to talk to you about. Former lab members, some of whose work I mentioned, collaborators, and funding sources. With that, I'm sorry, I think I went quite a bit over time, but I'll be happy to take questions, and thanks a lot for your attention.
Thank you so much. Great seminar. All those beautiful movies as well. That was great. I see maybe a question in the chat, and then several people have raised their hands. I'm just going to call on whoever has their hands raised in the order I see on my screen. And so I think first up is Tim. If you want to go ahead and ask your question.
Yeah. Hi. Thanks for this very nice presentation. So clusters of cell surface molecules have been seen for a long, long time. I think you showed Zap70 in really beautiful clusters. Is it known I mean, certainly the cytoskeleton is involved, but is there a more specific explanation? Does it have anything to do with the topography of the surface? Is there or is there some specific, kind of cytoskeleton assembly that they're associated with? It would be lovely to know how these clusters form.
Yeah, thanks. Great question. Sorry. Yeah, we've been looking at these clusters. I don't know what's going on. We've been looking at these clusters for a while, and there's been a lot of work on that. They are becoming, they've been studied also in cell-free system from Mike Rosen's lab, I believe. It's clear that there's a connection between the cytoskeleton and the signaling clusters. But I think in the context of the real cell, what exactly causes a single cluster to form is not fully understood. So you need the clusters to potentially have actin polymerization, and then you need the actin polymerization to allow the cell to spread further to then generate more clusters. We did observe in our studies on topography that it changes for example, the efficiency of cluster formation and the intensity, the cluster density. So I think both all these mechanical cues are also important in the type of clusters that are formed. So I don't know, I don't think I answered your question fully, but that's something that we've been thinking about.
Yeah, that's okay. I think it's just important to emphasize it's a very important answer in the field. So if somebody could answer that, it would be a big breakthrough. Thank you.
Great. Vivek, you're next on my screen here.
Yeah, thanks for the insightful talk. I wanted to ask about when you show the actin rings on different stiffness. So I wanted to know if those are actomyosin rings or just the actin rings, because actin rings go away once you inhibit the ARP23, they diffuse. But what happens to the actomyosin ring? Do they depend on the stiffness as well?
That's a really good question. I would have shown you I did not, but there is the actin ring formation. In these cells, we did not, we weren't able to get really good myosin transfection to be able to actually visualize it in live cells very carefully. But with fixed cells, we have seen that the myosin also forms a ring or a peripheral organization, similar to how I showed you in Jurkat cells, but maybe not as clear. But there's other work, for example, from John Hammer's lab and other labs that have shown also in cytotoxic T lymphocytes and primary T cells the formation of the peripheral ring as well as the myosin ring. And again, the myosin ring is in the form of these arcs, which are formin mediated. So
Yeah,
they get disrupted when you inhibit formin, whereas the peripheral actin ring gets disrupted. So there's some kind of a competition between actin between these two regulators of actin monomers.
I see. Just a quick follow up as well. So, do you think ICAM engagement is important for the formation of these rings, or will they form regardless of the integrin engagement?
Yes, yes, absolutely, and that has been shown that ICAM engagement does lead to increased enhancement of myosin recruitment and ring formation. Yeah, yeah.
Hmm,
Yeah.
I see. Thank
Great. Adam, you're next up.
Thanks. Yeah, really riveting talk. Thank you, Arpita. I have a lot of questions and comments, but obviously I can only ask one or two. Um, so I suppose one thing I wanted to ask was regarding the normal forces versus the in-plane forces. It's really hard to actually figure out the forces that the T cell is actually exerting versus the forces we observe on the gel, right? So we could just be seeing pinching, like lateral pinching forces to
Mhm.
the gel, which then results in a downward deformation, but it doesn't necessarily mean that the cell's pushing down itself, if that makes sense. So it's kind of an impossible thing to solve, but I was wondering if you had any thoughts on that.
Right. We were thinking about it more like the cell is pushing, and then that I forgot to point it out because you see, well, on the gel. So what we can see is the gel, right? And we can see kind of this crater form. And then there may be what we see is kind of this pinching up on the rim. So there will be some interplay between this pushing and pulling. And your question is the cell pushing? So we can visualize, and we are working on that to actually look at the granules, the serosome, as well as the cell membrane in this crater. So ideally, we label the cell membrane, for example, and then be able to see where the cell membrane is going. We do have actin labeled cells. Um, it's just been a challenge to have sufficient expression level to correlate. So our original goal was to correlate actin, forces, granules, myosin, all of this in this tour de force study. Of course, we're doing
Mhm.
limited. But that's a very good question, and we're trying to kind of address that to see what is the cell doing. Yeah, yeah.
Yeah, because I mean, definitely staining the membrane would be really insightful because in my mind, it makes sense from the CTL's perspective to get the target membrane as far away as possible to create space in the center of the synapse. Because then I think one of the open questions in the field is how the CTLs protect themselves against their own perforin mediated like pore formation. And so
Mhm.
maybe creating that space because it would be interesting to know if the T cell membrane follows that deformation or if it actually creates that a big pocket for the perforin to work
Yeah,
in.
that's a very good point as well. Like I think it is shown that there is a pocket, but more at the electron microscopy level. And so maybe with these we can figure out more at the microscopic level what is going on at that interface. Yeah, yeah.
Thank you.
And also while this where exactly is the secretion taking place? Sorry, I want to mention that. We just haven't been able to visualize it due to some technical challenges, but that will help us answer some of your questions.
I wanted, Cheng, before I let you sort of ask your question, just along those same lines, I mean, from the videos, it seemed like the synaptic release coincided very well with the in-plane force generation. Is that accurate? Is that what? And I think that agrees with what Morgan Hughes published, and he particularly showed a correlation between degranulation and LFA forces, right? I mean, I think that was the claim.
Yeah. So we haven't been able to, excuse me, image degranulation itself. That would be a next step for us. But what we see is the granules appear to be in the crater, kind of at the edge. And we're trying to correlate where along the edge. So we have that information, Adam, for example, that, is it up top over here or further down in the crater? We have that information. So we can correlate the granule intensity and depth with the curvature, for example, of, the cell membrane and kind of what leads to the release. And yes, Morgan and, Dan, their work has shown correlation between some of these topographic features on those particles and the granule release. And I think with this, we can get a more high resolved view along with the forces. Yeah.
I mean, the other advantage is you get this the force orientation, which I mean, as you alluded to in your talk earlier, there are these models suggesting that the lateral force to the TCR is what's really important, not the in-plane.
But the question is important for what? I think there's multiple things, right? Important for activation and then spreading, but then there may be other forces that are important for the next set of things. So I feel different forces mediated by different cytoskeletal components and motors are important at different stages. I had a slide of the timeline, which I don't seem to have shown, but yeah, completely agree.
All right, wonderful. Cheng, you want to ask your question?
Yeah, Arpita, beautiful talk. Thank you. My question actually relate to the last sentence you just said, which is that the cell skeleton generate forces, different time scale, etc. Right? So, initially, people was like, oh, cell pools and through integrins and then through other adhesion molecules. Now, I think I have yet to see a molecule that bind to an immobile ligand that does not pull on, the cell does not pull on. Maybe an example, I don't. Whatever molecule that we look at using tension probe, like Khalid developed, you saw forces, right? Now, they have different magnitude, they have different dynamics. So I'm just thinking, maybe it's a consequence of the cytoskeleton dynamics and the different forces that you measure through different receptor. Maybe it's reflection of how that receptor connects or link to the cytoskeleton. Or maybe, so I mean the cell don't pull on the molecule if the molecule are not ligated to an immobilized ligand, right? If you use a soluble ligand, they bind, they don't pull, right? So definitely there was a signaling that induced the cell to mobilize the cytoskeleton. And I guess you can, I'm just trying to get your opinion on these thoughts.
So your question is the cell is just generating a force no matter what. Is that kind of
once it's induced by some
once it's
signal.
induced?
There are many signaling molecules. Integrin can deliver a signal, right? T cell receptor can deliver a signal. Co-stimulatory molecule can deliver a signal, right? PD-1, it's inhibitory receptor, but it's deliver a signal that induce pooling on the PD-1, right? So, the question is that how specific are those forces, or the cytoskeleton dynamics just does its thing, and whatever connects to it experience that forces?
I think that's a really good question and interesting thought. We've been thinking about it a lot. What is kind of the chicken and egg problem, right? Like what is generating the force and what comes before that? Um, um, I think it's largely driven by the cytoskeleton, and there's differences in the forces depending on what leads to different types of cytoskeletal dynamics in that sense. Um, but are you saying, I guess, is your question that is the force itself specific for a certain type of linkage or not? Is that kind of your question?
So I think that you can see different magnitude and different dynamics if
Right.
you are measuring it through different receptors.
Mm-hmm.
We've seen that others have reported that. So that's not my question. My
Mm-hmm.
question has to do with whether or not that's a consequence of the different receptor have different cytoskeletal linkage
Mm-hmm.
that resulted in the manifestation of different
Absolutely.
measurements.
Yeah. I think that's highly likely that these are what we're seeing are manifestation of how these different sort of molecular level linkages binding then lead to different types of cytoskeletal dynamics. I think that is, that has to be, that is highly likely the case. And
Uh,
yeah.
that's comforting. I guess we think
Yeah.
a lot.
So
Yeah.
what we've also been finding that especially I think actin flows, I didn't get a chance to make a model properly. I think in 2 or 3 or 4 months we'll get there with some of our studies. But there's sort of this overwhelming evidence that actin flows appear to be just really, really important. And what somehow modulates the actin flows, the actin bundles, both in terms of forces as well as some of the some of the other polarization machinery we see. Of course, microtubules and microtubule motors and myosin also play a supporting role, but perhaps the pliability, the viscoelasticity of the actin, just very rapidly modifiable actin cytoskeleton seems to be playing a really important role.
Awesome.
Can I jump in, Chen? I have to jump in.
Ning, thank you so much for joining.
Thank
Last
you.
question.
I
Yes, go
think
ahead, please.
Chen, your question, I think we don't know. In short, we don't know the answer. I think your question can be, separated into different levels. Is single ligand binding, receptor or single actin, binding, or it's clustering, right? So Michael Xi's earlier work have shown that if you measure attractions, if it's multiple ligands, they cluster together, it's not 1 + 1 = 2. So they have different, you can strengthen, you can have much more traction force generation than if you just added, let's say, 2 or 3 ligands together. So that's one layer of question. So the other question you said, different connections with cytoskeleton, linkage strengths. I think nobody has any measurement on that in the live cells and comparing for different cells. That has something to do, actually, the more fundamental question is the mechanosensitivities of different types of cells. Is that different for different type cells because of biology, because of organizational, cytoskeletal organization, as the current speaker has been alluded to, or is it because of something else? We don't know. And to Tim Springer's question about clustering of different clusters, I think also may have something to do, we don't know, have something to do with phase separation, liquid phase separation. Because there are some recent data showing that some proteins that can cluster together. So we don't know yet. So there are a lot of unanswered questions. I think it's exciting, actually. I enjoyed the talk very much today. Okay, very good job.
Thank you. Thank you. Yeah.
Yeah,
And thanks
very
for
good.
pointing that out, that
Yeah.
one of the key questions has been what underlies this sort of mechanosensitivity. And while we are
Right.
focusing on the cytoskeleton,
Yeah.
I alluded to receptors and integrins, but the thing that we haven't talked about right now, for example, there could be mechanosensitive ion channels and
Right.
other
Yeah.
things
And
in
then that's it. Yeah.
our mind
Okay. Yeah. Okay.
that may also be important. Yeah.
But I think certainly cytoskeletal dynamics plays a very critical role here, as your data show.
Yeah.
Yeah.
Wonderful, wonderful. This was such an amazing a talk and a great discussion. We ran over quite a bit, but all for a good cause. Thank you again, Arpita, for the
Yeah.
lecture.
Yeah, thank you. And sorry for running over. I wasn't able to keep track of the time in the Zoom. But I see there's some chat questions. I know the time is running out. So I wonder if you'd be able to email me these questions.
Yeah. Unfortunately, the person who asked that question, at least the first question, I don't see that person in the Zoom. We can sort of take a screenshot perhaps of the questions and then forward these to you. I know we're recording, so we'll have a record of the questions. I think, Alicia.
So yeah, I should get a chat history once the recording is over. So
awesome.
Yeah, that would be great. And thank you again for inviting me and giving me an opportunity to share about the work from our lab. And great to interact on Zoom with so many of you. Thank you.
Great. Thank you.
you.
Thank you again.